Hello students, welcome to biology class. This video is in continuity with the previous video of chapter Transport in Plants. The objectives that we are covering in this video are transpiration, factors affecting transpiration, significance of transpiration, stomata and transpiration, structure of stomata, and mechanism of stomatal action. Starting with the topic transpiration. Plants lose more than 95% water absorbed by them and utilize less than 5% for their normal growth and metabolism. Hardly 0.2% is used in photosynthesis, while the remaining is retained in the plant during growth. Process of loss of water in the form of vapor from the aerial portions of the living plant is called transpiration. Now, types of transpiration. There are two main types of transpiration. First one is foliar transpiration. Loss of water through leaf surface is called foliar transpiration. Foliar transpiration is further of two types, stomatal transpiration and cuticular transpiration. Stomatal transpiration. It is transpiration that occurs through the stomata. It is responsible for about 90 to 97% of the total foliar transpiration. It is the most important type of transpiration. Cuticular transpiration. Water vapor is also lost directly from the outer walls of the epidermal cells through the cuticle. Cuticle is a waxy layer of cutin that covers the epidermis of leaves and stem. Cuticular transpiration amounts for about 3 to 10% of the total foliar transpiration. Cuticular transpiration continues throughout day and night. It should be noted that greater the thickness of cuticle, lesser will be the rate of transpiration. Desert plants have thicker cuticle to cut down transpiration. Cuticle is thus a waxy layer secreted by the epidermis on the two surfaces of the leaf. Next is lenticular transpiration. Lenticels are small openings in the cork of the woody stems, twigs and fruits. The amount of water vapor lost through lenticels is usually insignificant, approximately 0.1% of the total water loss. Lenticels never close, they remain open all the times. Next is factor affecting transpiration. There are many internal and external factors that affect transpiration. First, discussing external factors. Light. Light affects transpiration because it is directly involved in the opening and closing of stomata. Second is atmospheric humidity. Low humidity outside the leaf favors transpiration because in such a situation, vapor pressure gradient between the internal leaf atmosphere and the external atmosphere is very sharp. The vapor pressure of the atmosphere decreases with altitude just like atmospheric pressure. Therefore, high altitude plants often show xeromorphic adaptation to reduce transpiration. Next is temperature. The rate of transpiration increases with the rise in the temperature. This is because at high temperature there is more evaporation of water from mesophyll cell and therefore there is a greater saturation of the leaf atmosphere with water vapor. At the same time, a rise in temperature lowers the relative humidity of the air outside leaf. As a result, there is rapid diffusion of water vapor from the humid atmosphere inside the leaf to outer dry atmosphere. Next is wind. Wind reduces the steepness of vapor pressure gradient and the rate of transpiration is also reduced. Breeze, that is the gentle wind, removes humid air present around the leaves and the rate of transpiration increases. Winds of a very high velocity of storms increases the rate of transpiration initially. Due to excessive loss of water, the guard cells become flaccid. As a result, the stomata close at a later stage and the transpiration stops. Atmospheric pressure. Lower the atmospheric pressure, higher is the rate of transpiration. At high altitude, the pressure is low and so the pine trees have a very higher rate of transpiration. Next is available soil water. In a dry soil, the soil solution becomes more concentrated. Therefore, there are fewer tendencies for water to enter by osmosis. It means there is less uptake of water by roots. Hence, the rate of transpiration is also lowered. Now, coming on to the internal factors that affect the transpiration. Structure of leaf surface. The rate of transpiration also depends upon the structure of the leaf surface. Presence of thick cuticle, wax layer or trichomes on the leaf reduces the rate of transpiration. Next is area of transpiring surface. Any decrease in leaf surface area will reduce transpiration and conserve water absorbed by plants. Many kinds of grass roll up their leaves during dry condition, thus exposing fewer surfaces to the air than do fully expanded leaves. Next is stomata. The number, distribution and structure of stomata greatly affect the transpiration. The greater the number of stomata per unit area, the greater its rate of stomatal transpiration. The distribution of stomata is also important. Next is stomatal transpiration. Structure of stomata. Stomata are the openings found in the epidermis of leaves and young stems. Each stoma is surrounded by two small but specialized epidermal cells called guard cells. The guard cells are connected to the adjacent epidermal cells through plasmodesmata. The walls are differentially thickened and elastic. Each guard cell has a dense cytoplasm with the prominent nucleus and numerous chloroplasts. Guard cells are modified epidermal cells that surrounds the stomata. The epidermal cells which surrounds the guard cells are known as subsidiary cells or accessory cells. Guard cells differ from the subsidiary cells as follows. Guard cells. Guard cells are kidney-shaped in dicots and dumbbell-shaped in monocots. Subsidiary cells, they are variable in shape. In guard cell, inner wall is thick and rigid, whereas the outer wall is thin and elastic. Subsidiary cells are homogeneous, thin-walled cell. In guard cell, chloroplast is present and in subsidiary cells, chloroplast is absent. This image shows the complete structure of stomata. Each stomata consists of a minute pore called stomatal pore or stoma that is surrounded by two guard cells. Guard cells are the only epidermal cells that contain chloroplast. The guard cells have unevenly thickened walls. The cell wall around the stoma is tough and flexible, and the one away from the stoma is thinner. The shape of the guard cells differ in dicots and monocots. Next, we are discussing number and size of stomata. The leaf surface, depending upon the species, may contain 1000 to 60,000 stomata per square centimeter. The size of each stomatal pore when fully open may measure 3 to 12 micro in diameter and 10 to 14 micro in length. On an average, fully open stomata occupy approximately 1 to 2% of the total leaf area. Next come distribution of stomata. Stomata are mostly found on the lower surface of the leaf but may occur on both the surfaces. Moving on to the next topic, mechanism of stomatal movement. Stomatal transpiration occurs when stomata are fully open. The opening and closing of stomata is direct response to increase or decrease the osmotic concentration of the guard cell. When the osmotic concentration of the guard cells increases, they absorb water from the surrounding epidermal cells and become turgid. Increased turgidity causes the outer elastic wall of the guard cells to stretch towards outside. The inner thick and inelastic wall also gets pulled along with it and assumes a concave shape. The stomatal pore between the two guard cells open in this condition. When guard cells lose water and become flaccid, the stomata close. Thus, stomata function as turgor-operated valve, as their opening and closing movement is governed by turgor changes of the guard cells. From this image, we can understand the opening and the closing of the stomata. In dicot plants, kidney-shaped guard cells are there. Their inner walls around the stoma are thicker than the outer walls. Now, when guard cells are distended by turgor pressure, guard cells expand. Now, the tough inner walls become convex and stomata open. And when the turgor pressure in guard cells decreases, guard cells become flaccid. Inner cell walls come closer and stomata closes. The turgidity of guard cells, which is regulated by their osmotic concentration, is the main cause of stomatal opening and closing. Various theories have been given to explain these turgor changes. Some of the theories are as follows. First one is photosynthetic production theory. According to the von Mohl, in 1856, chloroplasts in the guard cells synthesize osmotically active substances. These increase their osmotic concentration. The water from the surrounding cells enter the guard cells and thus their turgor pressure increases. This results in stomatal opening. The mechanism of this theory can be summarized as follows. In light or during the day, photosynthesis by guard cells take place. Sugar formation is there, which increases osmotic potential. Now, endosmosis occurs, which increase in turgidity. Now, walls of guard cells stretch and stomata open. Now, during the night, no photosynthesis occur by guard cells. So, no sugar formation take place. The sugar formed is used up in respiration. Now, there is decrease in the osmotic potential. Now, in cell sap, exosmosis occur. Guard cells become flaccid, walls relax and stomata closes. From this image, we can understand that during the day, due to the endosmosis, stomata become turgid, which results in opening of stomata. And during the night, due to exosmosis, stomata loses turgidity, results in closing of stomata. Next theory is starch-sugar hypothesis. This hypothesis was postulated by Lloyd in 1908, Loffield in 1921 and Sayre in 1926. This hypothesis postulates that during daytime, CO2 which released in respiration is utilized in photosynthesis of mesophyll cells. Therefore, concentration of CO2 around guard cells and neighboring cells reduced with rise in pH. High pH favors conversion of starch into sugars which get soluble in cell sap. In dark, CO2 is accumulated in guard cells as photosynthesis stops. It causes fall in pH of guard cells. At low pH, conversion of sugar into starch takes place. Guard cells become flaccid and stomata closed. From this image, we can understand the mechanism easily. In light, photosynthesis occurs. So, CO2 is utilized. This decrease in the CO2 concentration and leads to increase in the pH or protoplasm become alkaline. Now this leads to conversion of starch into sugar. This increased cell sap concentration. Now endosmosis of water take place which increased turgor pressure in guard cells and stomata open. On the other hand side, the reaction in the dark. No photosynthesis take place due to the absence of sunlight. There is increased CO2 concentration. That leads to decrease in pH due to formation of carbonic acid. Now, conversion of sugar into starch take place. That decreases the concentration of solute in the cell sap. Now, exosmosis of water take place. This leads to decrease in turgor pressure and stomata close. Next is active transport or potassium pump theory. The concept of potassium ion transport was given by Fujino. It was supported and elaborated by Leavitt and Reschke in 1975. It appears to be an active mechanism which needs ATP. It has been proved that accumulation of potassium ions bring the opening of stomata and loss of potassium ions the closing of stomata. So discussing opening of stomata during daytime. Opening of stomata depends upon following condition. First, presence of light that will decrease in starch content of guard cells and leads to increased concentration of malic acid in guard cells. Then there is influx of potassium ions in the guard cell and efflux of hydrogen ions from guard cells. Intake of chloride ions by guard cells. This leads to low concentration of CO2 in and around guard cells. There is high pH in guard cells. High turgor pressure in guard cell due to endosmosis leads to turgidity of cells. Turgor pressure more toward thin wall of the guard cell and stomata open. In the guard cell, starch is converted into malic acid in the presence of light during daytime. Protons thus formed are used by the guard cells for the uptake of potassium ions. Now this is an active ionic exchange and requires ATP energy and cytokinin. In this way, the concentration of potassium ion increases in guard cell. At the same time, the concentration of hydrogen ion decreases in guard cell. The pH of the cell sap in guard cells also increases simultaneously. There is also increased uptake of chloride ions by the guard cells to maintain the electrical and ionic balance inside and outside the guard cell. The malate anion formed in the guard cell are neutralized by the potassium ion. This results in the formation of potassium malate. In this image, we can see starch when combines with water, it is converted into hexarose sugar. Now this is converted into malic acid. Now malic acid then dissociates into malate anion and protons. Now these protons, they get exchanged with potassium ions. Malate anions when combined with potassium ion form potassium malate. Potassium malate enters the cell sap of the guard cells, thereby reducing the water potential while increasing the osmotic concentration of the cell sap. Hence, endosmosis occurs. Guard cells become turgid and kidney-shaped and the stomata opens. It is also observed that the CO2 concentration is low in and around guard cells during daytime. This is due to high photosynthetic utilization of CO2. It helps in opening of stomata. In this image, we can see the role of potassium and chloride ion and malate ion in stomatal opening. The ions accumulate in the vacuoles of the guard cells, lowering the water potential and thereby increasing the water uptake and subsequently opening the stomata. Next comes closing of stomata in absence of light. Now this follows the following condition. Closing of stomata depends upon following condition. First one, absence of light, that will decrease the concentration of malic acid in guard cells, and there is efflux of potassium ions from guard cells and influx of hydrogen ions in guard cells. This creates acidic medium of the cell sap in guard cells. This results in loss of chloride ions from guard cells. Now increase carbon dioxide concentration in and around the guard cells due to release of CO2, carbon dioxide in respiration combined with the absence of photosynthetic activity in dark. Presence of plant growth inhibiting hormone, abscisic acid ABA, and loss of turgidity of guard cells. All these conditions represents the reversal of the daytime events. Under these conditions, the guard cells lose water by exosmosis and becomes flaccid. This causes closing of the stomata. Now in this image, we can see the complete systematic diagram to explain the active potassium ion transport in plants. Next is role of abscisic acid ABA. ABA is a hormone that triggers closing of the stomata when soil water is insufficient to keep up with transpiration. Presence of abscisic acid favors closing of stomata by blocking uptake of potassium ions by guard cells in the dark. It also prevents efflux of hydrogen ions from guard cells. Abscisic acid and CO2 concentration together help in lowering the pH in guard cells and making the medium acidic. This helps in closing of stomata. Abscisic acid acts as stress hormone during drought conditions. Coming on to the last topic, significance of transpiration. Transpiration has been described as a necessary evil because it is not only vital but also unavoidable for plants as it works like biological steam engine. The stomata remain open for exchange of gases. This also results in the loss of water as vapor. Consequently, the water level in the soil is reduced, causing wilting of plant. The stomata, however, cannot be closed to prevent water loss because this would also stop gaseous exchange needed for respiration and photosynthesis. It has following significance. Transpiration help in the rapid upward movement of sap. The absorption of water and ascent of sap to various parts of the plant body is mostly due to transpiration. Transpiration pull is the upward direction and is responsible for the mass movement of water in an upward direction. Transpiration helps in absorption and translocation solute. Transpiration helps in the absorption and ascent of water. The minerals dissolved in water are also absorbed along with water. Transpiration also regulates plant temperature. It helps in dissipating this excess energy by evaporating water from the leaf surface and thus helps in keeping the plant cool. Some plants with a high rate of transpiration are able to cool their leaves by 10 degrees Celsius below the air temperature. Next, discussing antitranspirants. Antitranspirants are the chemical substances which reduce the rate of transpiration without affecting other physiological processes. Initially, colorless plastics, silica oils, low-density waxes, etc. were used as antitranspirants, but these did not prove to be good. Nowadays, phenyl mercuric acetate (PMA), abscisic acid, aspirin, salicylic acid are being used as antitranspirants. A good antitransparent increases leaf resistance but does not affect mesophyll resistance. Thank you so much. We will continue this chapter in the next video.